So in this video, we're going to be discussing the pyruvate dehydrogenase complex. So pyruvate is going to be the end product of glycolysis, and pyruvate is a very important molecule because it can enter multiple pathways inside of our cells. So pyruvate can be converted into lactate, which happens in the muscles and the red blood cells. Lactate can be then taken all the way back to the liver to be converted back into glucose via the Cori cycle. Pyruvate could also be converted into alanine, and this happens in the muscle when they break down protein for energy. This alanine then goes all the way back to the liver to be converted into glucose via something called the alanine cycle. Pyruvate could also be converted into acetyl-CoA by the enzyme pyruvate dehydrogenase complex. And when we get acetyl-CoA, it's what's going to enter the TCA cycle to produce more ATP. The last thing is that pyruvate could also be converted back into glucose by the process of gluconeogenesis. So in this video, we're going to discuss how pyruvate is converted to acetyl-CoA and it's entering into the TCA cycle, and we're going to discuss the pyruvate dehydrogenase complex. So, like we said, pyruvate is going to be converted into acetyl-CoA, and once pyruvate is converted into acetyl-CoA, it could enter the TCA cycle to produce ATP for this cell. Now, the enzyme that is responsible for this is called pyruvate dehydrogenase complex. Why is it called a complex? Because there are multiple enzymes in this complex. So basically what the pyruvate dehydrogenase complex is it's the stepping stone or the connection between glycolysis and the TCA cycle. So during the process of glycolysis, we get pyruvate, and this is going to happen in the cytosol. So the first step is going to be the transportation of pyruvate into the mitochondria, and inside the mitochondria is where pyruvate dehydrogenase complex is active. And of course, it's going to convert it to acetyl-CoA, where it could enter the TCA cycle. So let's talk in a little bit more detail about the pyruvate dehydrogenase complex. So like we said, it's a complex because it's made up of multiple enzymes. These enzymes are numbered E1, E2, and E3. So E1 stands for pyruvate dehydrogenase, E2 stands for dihydrolipyltransacetylase, and E3 represents dihydrolipyl dehydrogenase. Now, this is an enzymatic complex that is responsible for the conversion of pyruvate into acetyl-CoA. Okay, so now we're going to talk about the specific reactions that happens with the pyruvate dehydrogenase complex. So like we said before, the goal of this enzyme is going to be to convert pyruvate into acetyl-CoA that can be used in the TCA cycle. So let's discuss each step in more detail. So the first thing is that pyruvate is going to be combined with thiamine pyrophosphate, which is vitamin B1, and thiamine pyrophosphate is going to be the active form of thiamine. And it's going to form something called acetyl-thiamine pyrophosphate. And this is going to be catalyzed by the E1 subunit of the pyruvate dehydrogenase complex. And this E1 subunit is called pyruvate dehydrogenase. And of course, it's going to lose one carbon. So we're going to end up with 2 carbons here, and we're just going to add thiamine pyrophosphate onto it. So what happens here is that basically the pyruvate loses a carbon and thiamine gets added. The second reaction is going to be the interaction between acetyl-thiamine pyrophosphate with lipoic acid, and this is going to be catalyzed by the enzyme dihydrolipyltransacetylase, or E2, and this is going to be the second part of the pyruvate dehydrogenase complex, and it's going to form acetyl-lipoic acid. So basically what happened here is that we moved the thiamine and we just added lipoic acid. Now, lipoic acid is a very important cofactor in the pyruvate dehydrogenase complex. So how does the cell maintain adequate amounts of lipoic acid? And this is going to be done by the E3 subunit of the pyruvate dehydrogenase complex, which is called dihydrolipyl dehydrogenase. Now this subunit, or E3, is going to be responsible for maintaining the function of lipoic acid, and it's going to do so by consuming 1 molecule of NAD and 1 molecule of FAD+, and yielding 1 molecule of NADH. And this is how our cells maintain adequate levels of lipoic acid that is used as a cofactor in the pyruvate dehydrogenase complex. The last step is going to be the conversion of acetyl-lipoic acid into acetyl-CoA. And so what happens here is that we just remove the lipoic acid and we add a CoA to it, and we get acetyl-CoA. Now this acetyl-CoA, of course, is then going to enter the TCA cycle and yield more ATP for the cell. So the pyruvate dehydrogenase complex requires 5 cofactors. And these cofactors are going to be thiamine, FAD+, NAD+, coenzyme A, and lipoic acid. So if we go back to our reactions, we could see that we use thiamine, we use lipoic acid, we're going to use an NAD and FAD, and we're going to use coenzyme A. and these are going to be the cofactors that are necessary for pyruvate dehydrogenase complex to move forward. Okay, so we're going to discuss each of these cofactors in a bit more detail. So the first one is thiamine, or which is also known as vitamin B1. Now, thiamine is converted inside of our cells to thiamine pyrophosphate, which is going to be the active form of thiamine. FAD+, which is also known as flavin adenine dinucleotide. Now, this cofactor is synthesized from vitamin B2, which is also called riboflavin. So vitamin B2 or riboflavin is going to be added to an adenosine nucleotide to form FAD. So this is going to be our adenosine nucleotide, and what the cell does is basically it adds vitamin B2 onto it to form FAD+. Now, FAD is important because it can accept 2 electrons, so it can be converted into FADH2. So it's now carrying 2 electrons, and then it can go into the electron transport chain and produce more ATP. So the functions of FAD+ is going to be as a cofactor for some enzymatic reactions, and it also transports electrons to the electron transport chain. The third cofactor is going to be NAD+, also known as nicotinamide adenine dinucleotide. And what happens, so it's going to be synthesized from vitamin B3, which is also known as niacin. So what we do here is that we take an adenosine nucleotide, and then we add another nucleotide, but the nitrogenous base is going to be vitamin B3 or niacin. And this molecule is now called NAD+. So what are the functions? It has very similar functions to FAD. So it's a cofactor for some enzymatic reactions, and also transports electrons to the electron transport chain because it has the ability to carry an electron. So if it acquires an electron, it's gonna be converted from NAD+ to NADH, and of course this NADH can go to the electron transport chain and start synthesizing ATP. The fourth cofactor in the pyruvate dehydrogenase complex is going to be coenzyme A. Now, this is going to be synthesized from vitamin B5, also known as pantothenic acid. So pantothenic acid is just added to an adenosine nucleotide, and we get coenzyme A. The last cofactor used in the pyruvate dehydrogenase complex is called lipoic acid. Now, this is a cofactor for the E2 subunit of the pyruvate dehydrogenase complex. Now, an important clinical correlate is arsenic poisoning. So arsenic has the ability to bind to lipoic acid and inhibit it. And if you inhibit, so let's say someone consumes arsenic, so this arsenic represented by these red dots are going to bind to lipoic acid, and it's going to prevent the lipoic acid use in the pyruvate dehydrogenase complex, more specifically the E2 subunit of the pyruvate dehydrogenase complex. And of course, that's going to prevent the conversion of pyruvate to acetyl-CoA, leading to inhibition of the TCA cycle. So this is going to present as garlic-smelling breath, vomiting, diarrhea, coma, and death. So to summarize things up, now we're going to talk about the B vitamins' role in the PDH complex. So the first 4 B vitamins are used as cofactors for the pyruvate dehydrogenase complex. So vitamin B1, thiamine, is used. Vitamin B2, riboflavin, is going to be used to make FAD+. Vitamin B3, niacin, is used to form NAD+. And vitamin B5 or pantothenic acid is used to form coenzyme A. There is no vitamin B4. So to be able to memorize them, just know that the first 4 B vitamins are used in the pyruvate dehydrogenase complex as cofactors. Okay, so now we're going to talk about the regulation of pyruvate dehydrogenase complex. So the first thing that you have to know is that the dephosphorylated form of the pyruvate dehydrogenase complex is the active form, and the phosphorylated form is going to be inactive. Now, the enzymes that regulate this phosphorylation or dephosphorylation are going to be the PDH phosphatase, which leads to its dephosphorylation, and the PDH kinase, which leads to its phosphorylation. Okay, so what are the molecules inside the cell that affect the phosphorylation or phosphorylation of pyruvate dehydrogenase complex, and how does that affect its activity? Okay, well, the first thing is going to be if there's an elevated level of NADH to NAD plus to NADH ratio, it's going to lead to activation of the pyruvate dehydrogenase complex. Why? Because when you activate it, basically you're going to activate the TCA cycle and you're going to produce more NADH. So basically here, what you're telling the cell is that we need more NADH. When there's high levels of ADP, this indicates that the cell has low energy levels. So you're going to want to activate the TCA cycle. If there's calcium, calcium indicates muscle contraction. And every time there's muscle contraction, you want to activate the TCA cycle in order to produce more ATP. Now, the exact opposite of that is going to render the pyruvate dehydrogenase complex inactive. And this is going to be low NAD plus to NADH levels. This means that we have a lot of NADH levels. That means we don't need to activate the TCA cycle in order to produce more NADH. If there's high ATP levels, that means that the cell is in a high energy state, so there's no need to produce more energy by activating the TCA cycle. And the last one is going to be an increase in acetyl-CoA. Now, why does an increase in acetyl-CoA inhibit the pyruvate dehydrogenase complex? Well, let's say that the cell has a lot of ATP, and this ATP inhibits the TCA cycle. So the TCA cycle is like we have enough ATP. So this is going to lead to the accumulation of acetyl-CoA. Now, this accumulation of acetyl-CoA is then going to activate the enzyme pyruvate carboxylase, which is going to be the first enzyme in gluconeogenesis. So it's going to lead to the conversion of pyruvate to gluconeogenesis. Now, at the same time, it's going to inhibit the pyruvate dehydrogenase complex so that all the ATP that is produced from the TCA cycle is not wasted in making glucose. So one of these has to be active at a time. So when acetyl-CoA levels increase, they're going to activate the pyruvate carboxylase, and they're going to inhibit the pyruvate dehydrogenase. Okay, so now we're going to talk about a clinical correlate, which is pyruvate dehydrogenase complex deficiency. Now, this is an X-linked inborn error of metabolism in which a patient is born with low levels of pyruvate dehydrogenase. Now, this is due to a mutation in a gene called the PDH-A1 gene, and it's going to lead to low activity of pyruvate dehydrogenase. Okay, so like we said before, pyruvate has multiple pathways that it can take inside of the cell. So let's say that the cell has low levels of pyruvate dehydrogenase. That means that pyruvate cannot be converted to acetyl-CoA and thus cannot enter the TCA cycle, and that's going to lead to a depletion of ATP production inside the cell. At the same time, the low levels of acetyl-CoA are basically going to inhibit the activity of pyruvate carboxylase. Remember that the enzyme pyruvate carboxylase is only active when there's high levels of acetyl-CoA. So pyruvate is not going to be able to be converted into glucose in the process of gluconeogenesis. So pyruvate at this point could only be converted into either lactate or alanine. So how does this clinically present? Well, the clinical findings are basically a newborn who has poor feeding, growth failure, and developmental delays. The labs are going to show elevated alanine and lactic acidosis because all the pyruvate inside the cell is going to be shifted to either alanine formation or lactate formation. The treatment is simple. So these patients, we have to give them thiamine and lipoic acid. So these are the two cofactors that we talked about that are important for the PDH complex. So the thiamine and the lipoic acid are going to optimize the remaining activity of the pyruvate dehydrogenase enzyme that remains inside of the cells. The second important thing is that we have to start these patients on something called a ketogenic diet. Now, a ketogenic diet is basically a low carbohydrate, high fat diet. Now, the low carbohydrates are going to reduce the lactic acidosis. Why? Well, the less carbohydrates that you give, the less pyruvate that you produce, and the less pyruvate, the less lactate that you're going to produce. This is why these patients are given a low carb diet. The second thing is that we have to start them on a high fat diet and also on a ketogenic amino acid diet. Now, what does that mean? Well, a high fat diet means high levels of fatty acids. Now, fatty acids can be converted into acetyl-CoA inside the cell, and thus this acetyl-CoA can enter the TCA cycle to produce ATP and bypass the need for the enzyme pyruvate dehydrogenase. The second important thing is to start them on ketogenic amino acids, which are going to be leucine and lysine. And the reason is that basically these ketogenic amino acids can, from their name, produce ketone bodies. And one of the precursors for ketone bodies is going to be acetyl-CoA. So if we give them leucine and lysine, their cells can form acetyl-CoA that could enter the TCA cycle and thus produce ATP. And this is going to be the importance of a ketogenic diet in the management of these patients.